For the next lecture for Bio 412 Comparative Genomics, I'm going to be talking about the genomes of eukaryotes. Hopefully some of the main outcomes I hope for you all to take from this lecture are to understand the relationships among the major types of eukaryotes, or among a suite of major types of eukaryotes, and through the understanding be better able to appreciate some of the evolutionary paths from early organisms to mammals, as well as understand how comparative genomic approaches can be applied to the study of the evolution of complexity. So let's talk about eukaryotes. One of the reasons why we might be interested in the genomes of eukaryotes is to better understand, what exactly it is that makes a eukaryote. If we think about them, they're somewhere between 2 to 3 billion years old. They have some very specific characteristics that differ from prokaryotes. They have membrane-bound structures, we know as organelles. They have a true nucleus. They have linear chromosomes, not circular chromosomes. They have organelle structures, mitochondrial chloroplasts that are the results of an endosymbiosis event. They have a large amount of, we'll put large in quotations because it depends on the organism, but a large amount of non-coding repetitive DNA. And what we can see is that there looks to be, and historically we've looked at them as exhibiting patterns of increasing levels of complexity. Now, the order and nature of that complexity I think is up for debate, but as we compare them to other groups such as the prokaryotes, I think the idea that we do see increasing levels of complexity in some of these organisms is easily defensible. And over on the right here, we have sort of some of the main branches, and this is a non-rooted tree in this case, right? So in this case, it's not saying, where the base of this tree is to better understand the relationships between these organisms, but there are some possible proposed roots. We can see one sort of at the base of the tree here with the branches of Amoebozoa and Opistheconchs headed out in one direction. Another one down here with diplomads and trypanosomes as the other alternative base of this tree. So there are some different ways we can think about this. We don't 100% know how we might draw this tree, but if we're going to sort of cut down the number of species that we're going to be looking at and just take a look at some sort of the key ones for which we have become sort of the model organisms, or a suite of model organisms that we might, or representative organisms that we might consider from genomic perspectives, we can look at these suite of 8 species, right? And a lot of this, as I said before, like one of the benefits of taking this kind of perspective is by better understanding eukaryotic genomes and the evolution of these genomes, we can better understand ourselves as Homo sapiens. And so this is just a time tree that shows, the divergences of these different species on geological timescales. You can see that there are some sort of major events in terms of what's going on in the Earth, in its environment, through time. And you can see that as we think back to where the base of this tree for these organisms is around about 1.5 billion years ago, right? And that's the best estimate, give or take, or however many hundreds of millions of years. But that's about when we think there could have been a common ancestor, is projected to have been a common ancestor between organisms like Arabidopsis thaliana, which is a plant, and our whole other group of other eukaryotic species. So if we were going to go back to this tree, it would be round about somewhere in this part of the tree, back when there was some sort of common ancestor. So let's take a look at Arabidopsis thaliana first, as plants are definitely a lot different than this group of animal organisms we're going to be looking at later. So Arabidopsis thaliana, this is a plant organism. It's a plant genome. It's basically the fruit fly of botany, right? So this is one of the, if not the principal model species for plants. Plants were derived from what we think was a single cell ancestor, something that was likely similar to a modern green algae of some kind. They are currently dominated by angiosperms, or flowering plants, and Arabidopsis is an exemplar of that. With a relatively modest genome size of 146 megabases, or million base pairs. The Arabidopsis genome consists of approximately 26 and some change thousand protein coding genes, a small number of pseudogenes and alternatively spliced genes, and a relatively small amount of transposons. One of the reasons why Arabidopsis genomes are so interesting to us is and so useful to us is they do have this rather compact eukaryotic genomes. I mean, there are some plant genomes that are epically large, like some of the like loblolly pine and some of these other really big genomes. Arabidopsis thaliana is very different from that. It's got relatively short introns. It's got short intergenic regions. It's got high GC content in exons versus introns. It's very easy to identify, or straightforward to identify exons and introns. It does have a bit of a complicated genetic history. Plants are can be challenging because they often have experienced polyploidization events that can lead to large whole genome duplications, as well as we see evidence of local duplications affecting approximately 17% of the genes. So it seems like duplication is going to have a lot of effect on what we're looking at in Arabidopsis thaliana. Another common thing that we see in this genome, and we see in many plant genomes, is the chloroplasts seem a lot more gene dense than the mitochondria. If we think about this, though, as an outgroup to our animal organisms or our other branch of the tree, one of the things that's really interesting about Arabidopsis thaliana is that it has about 60% of its protein synthesis genes are homologous with those found in human. So there is actually a lot of stuff shared between eukaryotes in terms of gene sets. About 30% of the transcriptome of transcription regulation is also shared with humans. And one thing to note about Arabidopsis is that it has actually a lot more transcription factors than we do. So they have a much more diverse and complicated suite of tools to use, at least from a transcription factor perspective, in the regulation of gene expression. And another thing to note about Arabidopsis thaliana is it's a great species for looking at the role of genetic diversity and how it affects phenotypes. This is a highly diverse organism that occurs across a lot of different environments and exhibits a high propensity for local adaptation. So it's really a great species for studying the role of the connection between genotype, its interaction with environment, and how that can lead to phenotypic outcomes. Moving on from Arabidopsis, kind of our basal species to the animal taxon that we're going to be looking at, we're going to take a look at Saccharomyces cerevisiae, which diverged from these other animal taxon, more than 1 billion years ago. Last common ancestor between them. And of course, when we're talking about Saccharomyces cerevisiae, what we're talking about is yeast. Yeast is often looked at as one of the model eukaryote systems, and that's because it's thought to be relatively simple as compared to a lot of other ones. It's easy to grow, right? Like we can all grow yeast in our kitchens. It has haploid and diploid states, just like we would expect to see in a lot of our animal systems. You don't have to worry so much about all the crazy polyploidization effects that we see in plants. It's relatively safe to use and to keep around. It's not going to be pathogenic. And it's unicellular, so it's pretty easy to culture and to maintain. It was actually unicellular. Was actually something that evolved from a multicellular ancestor. As mentioned before, its genome is really compact. So if Arabidopsis is 100 close to 150 megabases long, yeast is about order of magnitude smaller, almost about 12 megabases long. Broken up into 16 chromosomes, about 3.5 times the length of an E. coli genome. But we can see that it's definitely more complex than a bacteria. It might have a lot of sort of similar ways of similarities, being this small organism though to bacteria. It actually can contain plasmids, which is interesting, but because it's got such a compact genome, there are actually very few repeats in their genome. There was a duplication event that I think has happened about 150 million years ago, and that was then followed by a series of gene loss. So it's got a pretty compact set of genes, somewhere around 6,000. There's some debate and back and forth about that number. There are introns, but few of them. So we can look at processes such as alternative splicing, where we're looking at these multiple exonic genes and how they're formed, but it's not like there's tons of them. Of these 6,000 genes, there's quite a few that have unknown function, of which quite a few of them have homology with prokaryotes. And we can see over here, some of the proteins that we've identified here. So lots of metabolism genes, all the sort of standard processes. And, one of the things that's sort of most interesting in yeast too is that, we can use this as a system. I think I mentioned this before, for studying gene function, because you can knock out a large number of these genes and the yeast will still be able to survive. So it's really interesting for us to be able to look and figure out what are the functional roles of these genes. Of course, yeast is important for us as an organism as well in its own right. Used in bread, beer, wine, biotechnology. If you like stevia, for instance, I think it's actually produced in. I think most commercial stevia is actually produced through inserting a gene into yeast and letting yeast produce it. So lots of ways that yeasts are important to us as an organism, outside of just using them as a sort of base for understanding how eukaryotic biological systems work. Next up, we're going to talk about Ciona intestinalis, which is a urochordate. So we're going to skip a whole bunch of intervening invertebrate taxa. We're not going to talk about fruit flies and C. elegans and lots of other important genomes, just because I only have so much time. So we're going to jump ahead and start to look at some ones that are a little bit more closely related to the human genome. So look at Ciona intestinalis, maybe diverged from the rest of the chordates around 700 million years ago. Here's a picture of one. Doesn't really look a whole lot like us, but if you look at their early developmental stages, they look very much like our early developmental stages. There's some classic drawings of these by Haeckel. These are thought to be relatively primitive chordates and indicative of organisms that might represent the origin of chordates and vertebrates. They've got a relatively small genome, also about the size of Arabidopsis thaliana, 160 megabases, and about 16,000 protein coding genes, of which about 60 of them have homologs to worms, to C. elegans, and or Drosophila. So they've got a lot of genes in common and sustained that are very similar to invertebrate genes, including a suite of them where it might be actually more similar to invertebrates than they are to vertebrates, even though they're more closely allied with us. Things like hemocyanin, which is similar to like what hemoglobin is for us. One of the ways that they're important, and we use them to study our developmental processes and better understand that. But these organisms are not thought to have adaptive immunity, so they're looked at as a good system for studying, how a system can evolve with just innate immunity. They do have some allorecognition processes, though, so there may be some immunity questions related to that are actually of specific interest. But they're kind of a nice outgroup for studying a system that does not have adaptive immunity, so that they can, it's like you've knocked out adaptive immunity in this case. So about a fifth of their genes have no homologs in other eukaryotes, and one of the things that is really interesting to see in these organisms is that if you look at Eukaryotes, sometimes you see that the differences in protein coding sequences are much greater among species. And we're talking between species, like various species of Ciona, than we see between humans and, say, birds. So a really diverse set of organisms, and it's interesting to think about what this means is because if we look at their evolution and divergence patterns, so this is looking at how divergent these proteins are for these organisms. These are all Eukaryotes, Salpintomyces, Bacillus slosseri, Ciona intestinalis, Ciona savigny, Oikopleurodioica, all these organisms, right? What we see is that there's really these organisms are actually diverging very rapidly from their most closely related taxa, which is the cephalochordate, Brachystoma floridae. And what we think that this indicates is that we see these rapid rates of evolution. We're not really sure why we see it. There could be a whole range of reasons why, but we do know that this group is evolving quite rapidly. So it's a great group for looking for how divergence in these proteins could be driving some of their biological patterns. So while Ciona gives us sort of our closest invertebrate perspective of what our ancestors may look like, we can start to move into some of the more vertebrate taxa and use them to understand some things beyond just sort of our relative early development, or perhaps how our immune system works. So if we look at fish, diverged a little more than 400 million years ago, maybe 450 million years ago. If we look at Tetraodon nigroviridis, this is the pufferfish. This genome is a pretty compact vertebrate genome, estimated to be about 340 megabases long. It does have some repetitive regions, but they're relatively few, and about 40% of its genome is actually coding sequence, which is a lot for a vertebrate. Got about 28,000 genes, which is more genes than what we have projected to have. And one of the nice things about this genome is that it's compact, so it's a little bit easier to look at, and it does a good job of sort of presenting the large structure scale of any vertebrate genome, right? There's considerable patterns of synteny with other vertebrates, and there's sort of large paralogous groups on different chromosomes. And one of the things that pufferfish have helped us specifically understand and identify is how fish are different than humans. And so we can also see from looking at their genomes that there's actually been a pretty big, there have been some significant genome duplication events that have happened in the history of fishes. And pufferfish is actually particularly helpful in this regard. So we were to compare the Tetraodon chromosomes with human chromosomes. This pattern shows sort of which ones are shared. So these are the Tetraodon chromosomes with the human colors painted on them, and vice versa, human chromosomes with the Tetraodon painted onto them. And what you can see in this is that typically when you're looking at this case, right, we would see that the human region aligns to two puffer regions, and so we can see that there are typically these puffer regions would appear in multiple places. Here, yellows here and here, pinks here and pinks here, so that they're not just located in one spot. And overall, what this, whereas vice versa, we see pufferfish regions, associated with mostly just one chromosome. We can see this a little bit better on this slide here, where when we zoom in on specific regions, say on human chromosome 16, we can see that one part of the chromosome is identified to be on chromosome 13 of the pufferfish, and other genes are located to be on chromosome 5. And what this indicates is that on chromosome 13, it actually has all of this whole synthetic region. It's just lost these genes in this spot, whereas on chromosome 5, it's lost these genes in these positions. And so this is a good indicator of how the pufferfish genome has these evidences of different suites of genes, but the same region of the gene on multiple chromosomes, indicative of a whole genome duplication. And it's actually really interesting for us to think about, because that means that when we have these duplication events, one of the hypotheses is that a lot of these genes, if they exist in functional purpose in other places, they can be lost in these other chromosomes because there's already a copy somewhere to take their place. And where we see genes that, and this is a regular pattern we see across this genome, where some genes are maintained and the ones are lost. And any place we might see like both of these genes be expressed or be retained, often that means that one of those genes has evolved to have a new function. So another really cool thing about taking this approach is that by identifying these components about what has been duplicated and where these large synthetic regions are, we can actually start to create what are ancestral chromosomes in this case. And so we can start to look back historically to see like what did the chromosome structure look like for organisms that would have been the common ancestor of fish and human. We can also just use that gene set, right, to better understand what their biology might have looked like as well. What about moving further down the tree? So now we're, inside and sort of the mid to late Paleozoic period. If we look at the genome of the chicken, here we're looking at the chicken, Gallus. This genome is, we started noticing the genome start getting a little bit bigger, which is 1.3 gigabases or 1.3 billion bases long. Approximately 23,000 genes spread across 38 chromosomes and 2 sex chromosomes. And the thing to note about birds is a lot of birds are going to be like chicken and have the ZW sex chromosome structure. And this means that instead of the case like with us, with humans, where we're XY, these ZW sex chromosomes mean that the female is actually the heterochromatic sex, not the male. There's a lot of, when we think about how chicken can help inform us about our genome, by looking at chickens, we can see that there's still like lots of regions of synteny between chicken and human. And there's actually much more synteny conserved between chicken and human, and that's these sections of genes that are organized together, than there is between mouse and human. So in some ways, they're important. They're a good proxy for us understanding what the role the ordering of genes can have. Only about 60% of the genes are homologous with human that we find in chicken. So 40% seem pretty unique to birds. And if you look at the relative level of similarity, there's about 80% similarity with rodent proteins as opposed to chicken proteins. And so, that may be also indicative of the fact that we do, we might have much more similar biology and physiological processes going on between rodents and humans than between chickens and humans. There are some parts of their genomes, however, that are well maintained. So, for instance, transport proteins are pretty well conserved between humans and chickens, whereas immune responses are actually pretty diverged with only 60% conservation. Not surprising, as the diseases we may be facing and dealing with are probably very different. Chickens also have some things in their genomes that are related to how specific their biology is. So, they have keratin genes that we don't have, and these are really important for for feathers, basically, and they lack stuff that you need to make hair, which makes a lot of sense. They also don't have proteins for things like milk and enamel and vomeronasal receptors, which also could be indicative of the fact that like smell may work differently for a lot of birds, and there may be a de-emphasis of the importance of these receptors in chickens, for instance. So let's move a little bit further down the tree. We can start looking at some other things that are a little more closely related to mammals, and we can look at the monotremes, and this bizarre distant cousin that we have, maybe separating from us, I don't know, around like 300 million years ago or something. This is the platypus, right? The duck-billed platypus, and this organism is a monotreme, which means that it has a single aperture for both its urogenital purposes and its digestive system. So, as a cloaca, which is basically the same place where it's going to excrete waste and do its reproductive activity. The duck-billed platypus, how it looks, it looks like a hodgepodge of something just kind of pasted together, and genetically and biologically, it goes way beyond the duckbill and the everything else, right? Just how it looks. It's just got so many unique things going on. It's a really amazing system just to think about how the evolution of novelty and adaptation works, convergence as well. So seen as sort of an intermediate between mammals and reptiles. It's got a ton of different of characteristics that it shares with mammals. It's warm-blooded. It's got hair. It has milk. It makes milk. It has a placenta and odor receptors that are very similar to mammalian ones. And then with reptiles, it's egg-laying. It lays eggs that have yolks in them, vitellogenin, which is very specific to fish and reptiles and amphibians, right? It's got no adult teeth, and it has venom. And then it also has these weird other adaptations, like it has electroreception. So just a very unique organism, and serves as both a model for homologous and independent evolutionary processes. If we just think of it from the reproductive perspective, it's got both yolked eggs, a placenta, and milk, right? So it's got all of these different ways, these different strategies that other organisms usually only use one to give nutrition to its developing young. Either yolks or a placenta, or and milk, which is not observed in all organisms. It's just a really interesting system. And then if you look at it genetically, it's got a crazy genome. It's got 10 sex chromosomes. 10 sex chromosomes. It's 10 XY, 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 right? It's got 10 sex chromosomes, but it's got no SRY. And SRY is kind of an important gene for sex determination. It's just missing in platypus for whatever reason. So it's got this really complicated sex chromosome system. The genome itself is about 2.2 gigabases and about 18,000 or so protein coding genes. And it's a really interesting organism to compare to humans and other placentals with, because it's got tons and tons of orthologs to placentals, right? So lots of genes that it shares with placentals, and even has sort of a very rudimentary placenta, different type of placenta than mammals, though. It's a yolk-feeding placenta. And then, just to add in other stuff, I mentioned this earlier, they have venom. So you think about how venom has evolved, right? They have actually evolved venom independently. So there's, a lot of venom has evolved from these beta defensin lineages. And we can see that there's this blue lineage, lineage 1, that has evolved, where venom has evolved in reptiles. And we can see that the venoms that have evolved in platypuses are of a completely different lineage than the ones in reptiles. So very interesting system. So these venom proteins too look to be relatively phenotypically similar, but so do the same thing, but have a complete independent evolutionary history. So really interesting organism to compare for a lot of different interesting biological questions. Next up, we're going to talk about Canis lupus, or dogs. These guys diverging around, I don't know what, like a 200 million years ago, 150 million years ago. So in this case, we have dogs which can kind of serve as an outgroup to humans. So we're starting to get a little bit closer to the human lineage. And as we get closer, we're going to see a lot more shared components with these organisms. And the more that we can observe and understand things that are going on in dogs, the better we can understand things that might be going on in us. They're great systems, and additionally for studying things specific to their lineages or other lineages that might be interesting to us, like the origins of domestication. If we think about how diverged they are from wolves, it's really only a very small part of their genomes. Only 0.04% diverged from wolves at an exonic level, and 0.21% in introns. So if we're looking for the genetic basis for domestication, they're an excellent organism to study, as we can see the differences between them and wolves. And then we can look for those same kinds of processes in things like cows and chickens and pigs and anything else that any sort of wild organism we might actually be interested in domesticating. There's some really interesting work on Arctic foxes here by Russians. One of the other really interesting things about dogs is they share many of the human genetic diseases that we have, and they're really prone, because of the way that we've bred dogs, to things that exhibit Feinberg's effects, where they have such a small genetic gene pool that they're less prone, or they're prone to exhibit some of these diseases or underlying conditions. They have incredible amount of phenotypic variation, both morphologically and behaviorally. As I'm sure you all know, dog breeds are bred for a specific purpose, and a lot of their behaviors are programmed into them. So we can really use this system as an excellent means of understanding the genetics of both morphological and behavioral variation. Their genome is similar in size to ours. It's 2.4 gigabases. They've got a lot more protein coding genes, perhaps as many as around 38,000, spread across 39 chromosomes. They've got a lot more signs than us, so short interspersed elements, but they share a lot of the syntenic blocks that we have, both with mice and human. And a lot of the genetic variation we see is specific. We see the most of the variation between breeds as opposed to within breeds. And we can map those differences to about 2.5 million SNPs. One thing that we do see within breeds is we actually see a lot of linkage disequilibrium, so large blocks of DNA that has to be co-inherited. And they're several megabases long, and this is actually maybe one of the reasons why they're so easily broken up into different breeds and bred and selected for so rapidly for these different characteristics. It's because they have these large sections of co-inherited DNA that may allow them to evolve quite rapidly if we can select on them. Here's a nice little question for why can't cats and dogs just get along, right? Well, they're very different lineages. So if we think about where they diverged from each other, diverging around 40 million years ago from each other, that's a lot of time. They've got very different makeup. We can see dogs are here along this tree, whereas the cats are up here. So they split off into wolves. Canidae split off into wolves, coyotes, jackals, and red foxes about 4 million years ago. And the first records for domesticated dogs around about 20 to 15 million years ago, with sort of modern dog breeds exploding between about 1,000 to 3,000 years ago. But the main diversification of dogs, it happened really only within, say, about the last 200 years. So really, what we think of as a dog is a pretty recent entity. And they are. We're going to talk a lot about them. We're going to read some other papers about them. They're just a really interesting set of organisms. Lastly, human was kind of the last branch on this tree, but when we think about like what we actually use to study human, typically we aren't actually looking at humans for a lot of the work that we do. We will in the future, and we do so more now because generating genome sequences have become so easy. But really what we've often used are other related taxa to better understand ourselves. So if we think about the role in science of a model organism like musculus or Rattus novechicus, right? Like these are the mouse and the rat, right? These are the experimental organisms that have become so important for us and that we use as really the model species for looking at ourselves. And the reason why is that, organisms like dogs, like rats, like mice are seen as being very physiologically similar to us, and perhaps less ethically complicated in terms of using as experimental animals, like things like chimpanzees. So if we think about mice, for instance, they diverge just a little bit from us, just a little bit after dogs, say around like 80 something, 90 million years ago, right? And in this case, what we're looking at, when we think about musculus, it really is the principal lab animal in the history of science, right? So one of the reasons why their genome is so important to us is because of their role here. And why mice? Well, they're easy to raise. They were everywhere. They have a high rate of reproduction. They breed in captivity. There's a lot of things that mice have that are very similar to humans. Not everything, but a lot of things. Last common ancestors thought to be around 75 million years ago or so, whereas mice and rats have diverged from each other probably around 12 to 24 million years ago. Their genome is comparable in size to ours, a little bit smaller, 2.7 gigabases. They've got 19 autosomes. They have XY chromosomes, just like we do. They have a similar number of genes, a few more, maybe around like 22,000 or so. And they have a lot of homology with us, as well as high similarity. And also a lot of conserved exon-intron structures between human and mouse. We do see some differences in specific gene families. For instance, like photoreceptors are more expanded in mice, which makes a lot of sense, as smell is probably more important to them than to us. One of the things that's really interesting when we think about mice is that, we see, if we look at patterns of synteny between humans and mice is actually, pretty broken up. We can see that there are large sections that are synthetic with humans in various places. But if we compare it to something like rat, which is a lot more closely related, we can see the patterns are very different. So we've got human on the right side of each one of the left side of each one of these bars, and rats on the right. And what we can see is that there's a lot more variation in these colors when it comes to humans, right? So there's been a lot more recombination between these regions. So if we look at actually a dot plot of it, what you see is something that's very indicative of that, where you see some large sections for each of these chromosomes, but then you see a lot of smaller stuff broken up, right? And so what this basically means is, if we think of things from a synthetic perspective, that things are co-located in the same places in the genome, and I spelled Synteny wrong right there, so just ignore that. its synteny with human is going to be variable, right? So it's 90% synthetic, but the block region, like how large those regions of synteny are, can often be quite small. But that hasn't stopped us from using it as a disease genetic model system for us, right? And so, there's about 1,000 genes or so, probably more, that have known mutations in humans that are homologous in rodents. These cover all different kinds of things: muscular dystrophies, craniofacial and skeletal abnormalities, Lou Gehrig's disease, blindness from cataracts, retinal degeneration, glaucoma, etc. And one of the really big benefits of this system has been using it to really understand how these genetic systems work. And we've done that through being able to genetically manipulate mice. Mice, again, are easy to breed in the lab, and they've been one of the main sources of focus for looking at things like genetic manipulations. And what we do in this case, right, is we can actually we can do a couple different things. We can add genes into a mouse and see what they do. We can knock out a gene in a mouse and see what we can do, and we can even knock a gene into a mouse and see what we can do. So there are a lot of different systems for doing. There's a lot of different ways that we might go about doing this, but some of the best known ones are systems like the Cre-Lox system. And if you want, you can go ahead and click on this link online, and you'll come to a nice little blog post about explaining how the Cre-Lox system works in a little more detail than I'm going to go over today. But what this Cre-Lox system is a way for us to do tissue-specific inducible knockouts, or knock-ins, actually. You can do those too, where you can use a Cre gene, which is a site-specific recombinase, that should be a B, not a V, recombinase enzyme. And that Cre recombinase will interact with this LOXP site. Right, so LOXP is just a site-specific part where the Cre recombinase will target. And what happens is you have an organism that will express Cre in, say, a specific tissue like liver, and you have some sort of gene that you're interested in targeting. And you have a LOXP mouse where you've genetically manipulated so that it has LOXP sites sort of before and after this gene X. And then you have a Cre mouse that has this liver-specific Cre transgene. So you've somehow added this Cre gene so that it's always going to be expressed in liver. It's always going to be expressing this Cre recombinase. And actually, this system is actually originating from a viral system, from a phage system. What happens is you breed these together, right, and you end up with one copy of your liver-specific Cre mouse along with the Cre-specific transgene and one copy of the LOXP genes. You then mate this mouse with another mouse, and now you've got two copies in about 25% of your offspring, right, to your little Punnett square. You're going to have mice that are going to be homozygous for both the floxed LOXP sites, as well as have the liver-specific Cre trans enzyme. And what happens here is now this Cre will interact with these two LOXP sites, and it's going to cut this gene out. And what that'll do is that mean now in the liver of this organism, there will no longer be gene X. And so what you're able to do is look and see, well, what is the effect of this gene when I remove it? And that will help tell you sort of what is the function of the thing. So we then look for various phenotypic differences to try to figure out, how it might be different. What are the things that it does to this biological system? But it helps us really understand what the disease phenotypes are in mice. Now, this isn't the only way to do this. CRISPR-Cas9, which you've heard quite a bit about, and I could explain it here, but to be honest, I'm probably not going to do as good a job as this well-produced TED Ed video. So I just thought I'd show you this quick video. It's only about 5 minutes long, but this would be a good example of how an alternative method, which is becoming a lot more widely applied to accomplish some of the same things that we have been doing in mice already.
From the smallest single-celled organism to the largest creatures on Earth, every living thing is defined by its genes. The DNA contained in our genes acts like an instruction manual for our cells. 4 building blocks called bases are strung together in precise sequences, which tell the cell how to behave and form the basis for our every trait. But with recent advancements in gene editing tools, scientists can change an organism's fundamental features in record time. They can engineer drought-resistant crops and create apples that don't brown. They might even prevent the spread of infectious outbreaks and develop cures for genetic diseases. CRISPR is the fastest, easiest, and cheapest of the gene editing tools responsible for this new wave of science. But where did this medical marvel come from? How does it work? And what can it do? Surprisingly, CRISPR is actually a natural process that's long functioned as a bacterial immune system. Originally found defending single-cell bacteria and archaea against invading viruses, naturally occurring CRISPR uses 2 main components. The first are short snippets of repetitive DNA sequences called clustered regularly interspaced short palindromic repeats, or simply CRISPRs. The second are Cas, or CRISPR-associated proteins, which chop up DNA like molecular scissors. When a virus invades a bacterium, Cas proteins cut out a segment of the viral DNA to stitch into the bacterium's CRISPR region, capturing a chemical snapshot of the infection. Those viral codes are then copied into short pieces of RNA. This molecule plays many roles in our cells, but in the case of CRISPR, RNA binds to a special protein called Cas9. The resulting complexes act like scouts, latching onto free-floating genetic material and searching for a match to the virus. If the virus invades again, the scout complex recognizes it immediately, and Cas9 swiftly destroys the viral DNA. Lots of bacteria have this type of defense mechanism, but in 2012, scientists figured out how to hijack CRISPR to target not just viral DNA, but any DNA in almost any organism. With the right tools, this viral immune system becomes a precise gene editing tool, which can alter DNA and change specific genes almost as easily as fixing a typo. Here's how it works in the lab. Scientists design a guide RNA to match the gene they want to edit and attach it to Cas9. Like the viral RNA in the CRISPR immune system, the guide RNA directs Cas9 to the target gene, and the protein's molecular scissors snip the DNA. This is the key to CRISPR's power. Just by injecting Cas9 bound to a short piece of custom guide RNA, scientists can edit practically any gene in the genome. Once the DNA is cut, the cell will try to repair it. Typically, proteins called nucleuses trim the broken ends and join them back together. But this type of repair process, called non-homologous end joining, is prone to mistakes and can lead to extra or missing bases. The resulting gene is often unusable and turned off. However, if scientists add a separate sequence of template DNA to their CRISPR cocktail, cellular proteins can perform a different DNA repair process called homology-directed repair. This template DNA is used as a blueprint to guide the rebuilding process, repairing a defective gene, or even inserting a completely new one. The ability to fix DNA errors means that CRISPR could potentially create new treatments for diseases linked to specific genetic errors like cystic fibrosis or sickle cell anemia. And since it's not limited to humans, the applications are almost endless. CRISPR could create plants that yield larger fruit, mosquitoes that can't transmit malaria, or even reprogram drug-resistant cancer cells. It's also a powerful tool for studying the genome, allowing scientists to watch what happens when genes are turned off or changed within an organism. CRISPR isn't perfect yet. It doesn't always make just the intended changes. And since it's difficult to predict the long-term implications of a CRISPR edit, this technology raises big ethical questions. It's up to us to decide the best course forward as CRISPR leaves single-celled organisms behind and heads into labs, farms, hospitals, and organisms around the world.
So you can see there's some really interesting systems and different systems that you can use to do genetic manipulations in mice, and it seemed to be a really powerful model for this. Most drug trials, experimental trials, all go through some sort of mouse stage. But there are lots of things where mice are just not the same as us, right? So there's lots of diseases that may be similar but are not exactly the same. For instance, a lot of mouse cancers don't look anything like human cancers. So maybe not the best system always to study human cancers. But still a really powerful, really useful system, one that gives us incredible insight to humans. There are issues, of course, like, mice are different sizes than us, so their metabolism and their physiology can be really different than us. Their life history is really different from us. Their diet, their microbiomes, pathogens are all different than what we'd experience. And, what we're looking at when we look at mice are really a very diverged, domesticated strain of mouse as opposed to what the wild version is. So there are some reasons why mice can be challenging as a comparative system to us, but they still are an incredibly powerful and useful model system for looking at how biology very similar to ours can function. So these are just a bunch of examples of different types of genomes and how you can see the process by which complexity has built through. We've gone from sort of these simple yeast systems with small genomes and few genes, 12 megabases, 6,000 genes, up to the mouse, which is going to have a similar genome size to ours, 2.7 gigabases, and similar number of genes, around like 23,000, right? But we've seen that, the systems have evolved to have lots of different idiosyncrasies and specific things to each of them. But it's by looking at the things that we share and the things that are different between these different organisms and their genomes that really is going to give us a lot of insight into what the true, into our own biology and the biology of other organisms and sort of the broader rules that govern how biological systems work. So, as a general introduction to eukaryotic genomes, hope this has been helpful, and look forward to our next lecture where we'll be discussing the intersectionality between genomics and anthropology.